Namaskar. So in this video we will discuss blood grouping system, right? Before discussing this blood grouping system in detail, we have to clear the few common terms that we will use everywhere, that is antigen and antibodies. What are the antigen, students? Antigens are antigens are actually, they are chemical substances, right? And once they enter in body, they instigate production of antibodies. Yes, they are chemical substances which provoke production of antibodies. Okay, now these antigens, uh, they can be protein. Mainly the most powerful kind of antigens are protein, or they can be some kind of lipopolysaccharides also. Lipopolysaccharides also. Okay, or they can be even carbohydrate also. They can be some kind of sugar also, right? But they cannot be fatty acid and all something. Fat they cannot be. So antigens can be lipopoly. Mainly they are protein. They can be lipopolysaccharide, like carbohydrate. Like like you see, we have one bacteria, okay, that entered in your body and causes production of antibody. That means the cell wall of the bacteria have lipopolysaccharide. The different parts of the body of that bacteria have different proteins. They are only instigating production of antibodies. Now these antigens are two types, students. One is self antigen, another one is foreign antigen. What are the self antigen? See, you have a body have cells, and cells have your own specific proteins. Like you see, in your body you have some particular proteins in your cell, right? That is species specific. In same way, in every species there will be some specific protein. So in your body, your proteins are self antigen. Remember, students, body will never produce antibody against self antigen normally. Normally they will never produce antigen against against antibody against self antigen, right? But body immediately, once they recognize foreign antigen, invader, some kind of antigen, they came from outside to inside, then they will produce antibodies, right? How they will produce antibodies? Try to, they actually, they produces this antibody, they feel that is some kind of threat for me, right? And it's going to damage my body, that's why they produces, right? So question is that how they produces this antibody. Try to understand. Like, I have one antigen that entered in our body. Antigen, it's a globulin protein, right? And the motive of secretion of this globulin protein, which is to provide immunity. That's why antibodies are also called immunoglobulin. Immunity, protection. Immunity means protection, right? So immunoglobulin, right? So antibodies are also called immunoglobulin. Like, see, in our bodies some kind of antigen entered, right? So once antigen when entered, the pillar of our immunity, major pillar of our immunity, B lymphocyte, will recognize it. And this B lymphocyte, according to shape, size, structure of antigen, will differentiate into plasma cells, and these plasma cells will start to secrete ample of amount of antibodies according to shape and size of antigen. Okay, see, if antigen is second type, so they will produce second type of antibodies. If antigen is third type, fourth type, fifth type, million type, so they will produce third type, fifth type, million type of the antibodies. N number of antigens we have, n number of type of antibodies will produce, right? How it will produce? By plasma cell. How? From where this plasma cell came? Plasma cells actually is differentiated lymphocytes, right? B lymphocytes. And this B lymphocyte will differentiate only when some kind of foreign antigen enter in body. I hope this point is clear to you, that what is antigen and what is antibodies, right? Now, on the basis of some self antigen, we have divided our blood into different blood group. Yes, on our red blood cells in our body, in our red blood cells, on the surface of red blood cell, in the plasma membrane of red blood cell, some antigens are present. Obviously they are self antigen, isn't it or not? They are self antigen. On the basis of that, we divided our blood group in our blood into different group. Karl Landsteiner have given first blood grouping system, very important, ABO blood grouping system. In 1900, Karl Landsteiner, when he was studying red blood cells of the human, he observed that on the red blood cell of the human, some specific type of antigens are present. Okay, and that antigen, on the basis of antigen only, he given this blood grouping. If A antigen was present, he told that okay, the person had A blood group. A blood group. If it have B antigen, then it told okay, this guy have B blood group. B blood group. And if both and B antigens were present on the some people's red blood cell, he told that okay, this guy had AB blood group. This guy have AB blood group. Okay, those who have A antigen on their red blood cells, A blood group. Those who have B antigen on their red blood cells, B blood group. Those who have AB antigen on their red blood cells, AB blood group. And those who have neither A nor B, they have actually O blood group. Yes, O blood group. Then they studied plasma of the individuals. When they studied plasma of individual, they completely shocked. They observed that nature have given antibodies also against it. Naturally, in our body, antibodies are present. Those who have A antigen, they have B antibody. See, those who have A antigen, they have B antibodies. Yes. Those who have A antigen means those who have A blood group, in their plasma B antibodies are present, not A antibodies. See, if A antibody will be present in A blood group peoples, immediately that A antibody will come, they will bind here and they will hemolyze the RBC, they will break the RBC, right? So this guy, for protecting their blood from B blood group, nature have given B antibody. Okay, right? Those who have B blood group, they have B antigen, nature have given them A antibody, right? So A antibody will present, they will not react to their red blood cell, and if in their body, if those who have B blood group in their body, if A blood group will enter, then this A antibody will kill that all red blood cell, they will remove the all blood cell, they will eliminate that all antigens, right? And those who have AB blood group, they have A and B antigen both, so they have neither A antibody nor B antibody. And those who have neither A or nor B antigen, that means those who have O blood group, they have AB antibodies, right? So AB antibodies present in O blood group. In AB blood group, no antibodies are present. Those who have B blood group, A antibody. Those who have A blood group, they have B antibody. Clear? Clear, students? Now, question is that what kind of situation will create, hello, students, what kind of situation will create, okay, actually, blood transfusion, is very common process in medical science. Like person is going through some kind of surgical operation, right? He got the road accident, hit and run, someone hit and then he run. Okay, so in that situation, lot of blood transfusion is required. Now it's very important to decide that which kind of blood group should be transfused to whom, because we have to consider this antigen, antibodies and all, right? How it will be decided, that is very important. Try to understand, students. See, first point, remember, for blood transfusion, same blood group will be always prefer. Like I have A blood group should prefer to me. I have B blood group should prefer to me. I have AB blood group, then AB blood group only prefer to me. O blood group should prefer to me, right? I am taking one situation. I have suppose one great doctor. Anyone? Okay, he's great doctor without degree. Okay, don't take it personally, but there is one person, his name is Costume. Okay, he need blood transfusion. He went to the doctor decided to transfuse the blood, right? But this Costume have A blood group. Okay, and the A blood group individual was not available, so he approached one person who have B blood group, and they transfused B blood group into the Costume who have A blood group. Donor had B blood group, acceptor had A blood group, right? Now what kind of situation created, you see? See, if this guy acceptor have A blood group, that means they have A antigen and they have B antibodies all over their body. Now the blood which was transfused here, that is B blood group, in that means in their red blood cell B antibodies are present. Now this B antigens are present, isn't it or not? Sorry, it should be B antigen. B antigens are present, yes. Now this B antigen containing donor red blood cell enter into the body of the person who have A blood group. Now this B antigen was completely alien to the body, foreign to the body. Immediately B lymphocytes activated and they differentiated into plasma cells, right? Because they seen one foreign antigen, right? And then plasma cell produce lot of B antibodies. And these B antibodies, unfortunately, they binded with B antigen of red blood cells and they hemolyze them. And it's happened some kind of mass level. Now this mass level of destruction of red blood cells causes shivering, and when red blood cell break, hemoglobin also break, and when hemoglobin break, lot of bilirubin form, which is quite poisonous substance, and that will damage the kidney, that will damage the liver, and that will can damage the brain also, and it can cause death. So what I told, wrong blood transfusion can lead to even death also. Why? Because when in wrong blood transfusion, different blood transfusion whose antigen is not present in body, that body will immediately instigate production of antibodies by the plasma cells. They will bind to the donor red blood cell and they will hemolyze them. I hope this point is clear to you all, right? So wrong blood group should not be transfused. Now come to concept of universal donor and universal acceptor. Try to understand, students. See, I have one person who have AB blood group. You see, this guy have AB blood group, right? Now this guy have AB blood group. That means in their blood, A antigen also present, B antigen also present, isn't it or not? Obviously, they can accept blood from AB blood group, isn't it? And they can accept blood from even B blood group also. Why? Because B blood group have B antigen, right? That is already present in their body. They can accept blood even from A blood group also, because A blood group have A antigen that is already present in their body, so it's not going to harm them. Okay, and then O blood group also they can transfuse him. Why? Because O blood group have neither A antigen nor B antigen, or even this also will not going to harm that person, right? So this person can accept from blood from A also, B also, AB also, O also. Universal donor, see, is drinking okay, is drinking blood from all kind of, obviously, during blood transfusion will not allow person to drink the blood, will transfuse intravenously, right? So yes, so but This person can accept blood from A blood group, B blood group, O blood group, and AB blood group. So that's why we'll call this guy universal acceptor. See, this universal acceptor, universal donor, these all concepts are valid only for 12th level, 11th level, right? Or NEET exam. In lab, these all concepts have no value, right? Because in lab, during blood transfusion, we take blood from donor, we take blood from acceptor, and we mix it. If blood is after mixing, they are forming clump, they are aggregating, that will not allow blood to transfuse, even they are same blood group then also. Okay, yes. Now come to concept of universal acceptor, students. Universal acceptor, see this guy is universal donor, B blood group. Why this guy is universal donor? You see, this guy can donate their blood to A blood group also. Why? Obviously it will transfer blood to, they can donate blood to the O blood group, right? They can even donate to A blood group also. Why? Because A blood group have A antigen, they have no antigen. So when it will enter in their body, this blood will enter in the body, it will fulfill their requirement, and they are not going to instigate formation of any kind of antibodies, because they have not any kind of foreign antigen. And then B also they can transfuse, and then AB also they can donate. That's why we will call them, universal, we'll call them universal donor. See this guy, after donating blood again and again, it see such is became such a lean. Actually it's not right, See, whenever you donate blood, it's not like that doctor will take your all 5 liter of blood. 350 to 650 maximum, this is what blood they will take. Okay, right? More than that ml they won't take. Reason is that your life will come in threat, and this volume of blood, you will your body will form within 24 to 48 hours, right? So blood transfusion, blood donation of blood, is completely uh, that process, a safe process. Okay, now come to RS blood grouping system. RS blood grouping system was developed by in 1940 by Karl Landsteiner, same person who developed ABO blood grouping system and AS Wiener. They observed that in the red blood cell of the human, 80% of the human, one antigen present, right? That antigen present even in the rhesus monkey also. Okay, so those who have Rh antigen, we given them a term Rh positive, and those who have not that Rh antigen, Rh negative, right? Okay, now those who have Rh antigen, obviously in their plasma Rh antibody is not present. If it will be present, then it will bind with their Rh antigen and hemolyze their RBC only, so it's not present. Even those who have Rh, those who are Rh negative, in their blood also Rh antibodies are not present. Until unless in their blood some Rh antigen will not enter. Try to understand it. This person is Rh negative. That means in their body Rh antigens are not present, right? But wantedly, if you transfused some red blood cell who have Rh antigen, then there immediately in their body B lymphocytes will activate. They will differentiate into plasma cells, and this plasma cell produces lot of antibodies. Which antibodies? Rh antibodies. And this Rh antibodies will eliminate everything. Clear? This point is clear to you all? So Rh positive, Rh negative, normally in their blood Rh antibodies are not present, but in Rh negative individual, if from outside Rh positive blood will enter, then their body will instigate formation of Rh antibodies. I hope my point clear to you. Okay, now come a little genetics of ABO blood grouping system. See students, actually this ABO blood grouping system present into the chromosome ABO locus. ABO locus is the region of the chromosome where alleles of ABO blood groups are present, and they have three allelic form: A, IA, IB and IO, right? Three allelic forms are present. Now from three allelic form, total how many combinations are possible? You tell. 9 combinations are possible or not? You see, like I'm taking father and I'm taking mother, right? If father sperm have IA, mother ova have IA, then zygote have IA. This guy have A blood group. This guy have A blood group. Okay. As A and B allele are codominant. Codominant means they are not dominant or recessive for each other. So if father have IA, mother have IB, then guy have AB blood group, right? And IO, you know, is recessive to A and B. I am repeating again. This IO allele is recessive, right? So IA and IO, A blood group. Okay. Same thing is here also, AB blood group, and here B blood group, here B blood group, because IO is recessive to IB, right? Here A blood group, here B blood group, and here O blood group, because they are recessive now, homozygous recessive now they are, so they are O blood group. So I hope now you got which kind of allele will be present into AB. I mean, the which kind of allele will be present in A blood group, B blood group, AB blood group, and O blood group. Before completing this session, you know, one very interesting point we need to know. See, those who have A allele in their body, A antigen will only form. Why? Those who have B allele in their body, B antigen will only form. Why? Those who have AB in their body, both A and B antigen will form, and those who have O allele in their body, no antigen will form. Why? Try to understand it. Before going there, we need to understand structure of this all AB antigens. See, for A and B antigen, their precursor is H antigen. Okay. This H antigen present in A blood group, primitive A blood group also present. Okay. This H antigen was precursor for B also, and H antigen was precursor for also O blood group also, and obviously for AB blood group also. Now see the structure of this H antigen. It have one glucose, it have one galactose, it have one N-acetyl glucosamine, one another galactose, and then one ficose present. Glucose, galactose, N-acetyl glucosamine, ficose present. This is H antigen, right? Now the H antigen was formed during embryonic development of our body, when blood groups were developing in our embryonic life, and the person had H allele. Sorry, the person have IA allele or IA allele, or person have IO or IA allele. So obviously in their body A antigen will form, isn't it or not? Because this allele will secrete some enzyme. That enzyme is called glycosyl transferase, and that will cause formation of A antigen, right? Now this A antigen, how different from the H antigen? You see, glucose is common, galactose is also common, N-acetyl glucosamine is also common, galactose is also common, ficose is also common, ficose is also common, and then that IA allele added one antigen, added one sugar, that is N-acetyl galactosamine. This enzyme, galactosyl transferase, they will add one N-acetyl galactosamine, then it will change, H antigen will change into the A antigen, right? Then if allele is either IO, IAIB or IBIB, then what they will do, then they will add another galactose here. You see, in H antigen they will not add N-acetyl galactosamine, they will add another galactose. See what is difference from the H antigen? Glucose is common here also, galactose is common here also, N-acetyl glucosamine is common here also, galactose is common here also, fucose is also common here also, fucose, sorry, not fucose, fucose, fucose is common here also, fucose is common here also, and then galactose present. Okay, galactose is added, so it's B antigen, right? So I hope now you got difference between A and B antigen structure. Those who have AB, that means they have A antigen, B antigen also, and those who have O blood group, those who have O blood group, right? Those who have O blood group, in their body H antigen will remain, in their blood group H antigen will remain. So remember, if in with context categorically it will be asked, so you have to tell in O blood group H antigen present. So in this video we discussed structure of antigens, then we discussed which kind of alleles are present in these antigens, and then we discussed universal donor and universal acceptor concept, and then we discussed how to transfuse blood, and then we discussed different antigens present into the ABO blood grouping system and RH blood grouping system, and then we discussed what will happen if a wrong blood group will be transfused, and then we discussed the concept of self antigen, foreign antigen, and then we discussed how antibodies produces. Remaining we'll see in next video. Bye.